Good evening, everybody, and welcome to this installment of the Young Scientist webinar series. Tonight, we'll be hearing from Hannah Wellman on marine mammals before extirpation, using archaeology to understand Native American use of sea otters in Oregon prior to European contact. And next month on December 14, we'll be hearing from Michelle Bautran-New Wang on Thinking Pink, investigating the impacts of ocean acidification and warming on larval Oregon pink shrimp. I also want to take a moment to acknowledge that the Cape Perpetua area landscape from Yachats to Florence is the traditional territory of the Siletz tribe and the Coos, Lower Umpqua, and Siuslaw tribe. And we want to acknowledge that the tribal governments and their roles historically and today in taking care of these lands. A little bit about the Cape Perpetua Collaborative. My name is Tara DuBois. I'm the communications coordinator for the collaborative, and it's my pleasure to coordinate this series. And the collaborative's vision is to foster conservation and collaboration within local communities for scientific exchange, management, awareness, and stewardship from land and sea in and around the Cape Perpetua Marine Reserve. Our three guiding principles are community engagement, leveraging resources, and engaging in partnerships. You can see the logos at the bottom here. These are the founding partners of the Cape Perpetua Collaborative, but we also have a variety of other partners, including local businesses, local agencies, local governments, and just general public and folks in general. We have so many volunteers that come out and support our projects. So we're just very grateful for the partnerships. A little bit, as I mentioned, the focus is around the Cape Perpetua Marine Reserve. It stretches from Yachats to Florence. It's Oregon's largest marine reserve, and north of the Marine Reserve and south of the Marine Reserve also include marine protected areas as well as a seabird protection area. So you can see the map here on the left gives you some kind of focal points of where the boundaries are. And some beautiful images of the area to the right, but you can find out more information about this on our website. We also host a variety of community science at Cape Perpetua Collaborative. You can see here the list of the projects that we host. Some of these are seasonal and some are monthly, which would be the monthly beach cleanups. We also host on Saturday mornings during the winter a Cape Perpetua speaker series. And we have our Land Sea Symposium coming up on November 19th, and we've got a really great lineup. So take a look at our website, capeperpetuacollaborative.org. If you click on the events tab, that'll take you to our calendar. And we will post all of our upcoming events, community science presentations there on that calendar, as well as on our Facebook page. And we will host replays of our presentations and as many events as we can that we film on our YouTube channel. And if you're inclined to support the collaborative, to keep projects like this going, do feel free to donate. There's a donate button at the top of our website. You can click that and that'll take you through the process. And with that, I'd like to introduce our speaker, Hannah Wellman. Hannah recently received her Ph.D. in Anthropology from the University of Oregon. Her research interests include past environments, human, animal, and human environment relationships, and ecological sovereignty. She is a Rising Sea Grant John A. Naus Marine Policy Fellow. And with that, Hannah, I'm going to stop sharing and you can take it over.
Great. Let me get my screen share going.
And while Hannah's bringing that up, I just want to mention that throughout her presentation, if you have any questions, please feel free to drop them in the Q&A section, and we will do a Q&A after her presentation.
All right, can everybody see my screen? Maybe Tara, can you see my screen? Perfect. I'm going to turn off my video so you don't have to watch me while I talk. All right, good evening, everybody, and thank you, Tara, for that introduction. I'm so happy to be speaking to you this evening. As Tara said, I finished my PhD last June, and I studied, as the title says, marine mammals before extirpation by using archaeology to understand Native American use of sea otters prior to European contact. And I also looked at pre-contact whale use, but in the interest of time tonight, I will be limiting my discussion to our favorite cute sea otter critters. So in terms of a talk outline, I'll first take you through some relevant background and context to get us all situated and on the same page. And then I'll go through my research and results. And then, as Tara said, we'll be taking questions at the end. So first off, some relevant background, although I realize I am probably preaching to the Oregon choir here. The Oregon coast has been home to tribal populations since time immemorial and is home to tribal populations today. It's a rich marine ecosystem with adjacent terrestrial resources. European contact occurred around the 1700s, and Euro-Americans overexploited marine mammals, particularly fur-bearing mammals like sea otter or beaver. And they drove some species close to extinction while inflicting disease, violence, and displacement upon indigenous communities. And I am generally interested in sea otters as well as cetaceans because these are animals that are subject to modern conservation concerns. There's usually sort of a robust public interest in these animals that's sort of part with this emotional component. These are very charismatic mammals. I know I personally have a very strong emotional response to, say, a whale beaching. And sea otters and cetaceans are also mammals which indigenous peoples have permission to hunt elsewhere on the Northwest coast. So I was interested in documenting the use of these animals as it might relate to ecological sovereignty and sort of arguing for tribal rights to sort of manage and or hunt these animals. So the archaeological sea otters that I studied and I will discuss in this talk come from two archaeological sites, the Partee and Pomro sites, and they are located in northern Oregon. And they're sort of just north of Tillamook Head, and they're south of the mouth of the Columbia River. And Partee is the most recent, dating to around 1800 to 1100 years before present. And then Pomro's is slightly older, dating 2700 to 1500 years before present. And because Partee and Pomro's are located so close to each other, they're sometimes also referred to collectively as the Seaside sites. And I'll refer to them as that through the talk. I should have mentioned they're located in Seaside, Oregon, hence the Seaside sites. Both of these sites were excavated in 1967 through 1977 by Robert Drucker and George Phoebus. And the animal remains from these archaeological sites are curated at the National Museum of Natural History in Washington, D.C., and the Museum of Natural and Cultural History on the University of Oregon campus in Eugene. And the Seaside area at contact was home to the Clatsop and Tillamook, and their descendants today are represented by Chinook Nation, the Confederated Tribes of Clatsop and Nehalem, the Confederated Tribes of the Grand Ronde, and Confederated Tribes of Siletz Indians. So why a research interest in sea otters? Sea otters were driven to near extinction in the 19th century Euro-American fur trade. They previously ranged continuously along the Pacific Rim from Japan all the way down to northern Mexico. But as you can see in this map, they are now restricted to these orange shapes. So parts of Russia, Alaska, British Columbia, Washington, and California. They're glaringly missing in Oregon. And as a result of the sort of severe population depression that came from that over aggressive hunting on the Northwest coast, for purposes later in the talk, I'll sort of be distilling this largely into an Alaska and California subpopulations of sea otters on the Northwest coast. Reintroductions to Oregon failed in the 1970s, so sea otters are technically locally extinct in Oregon. You can see them in the zoo, but those are Alaska sea otters. Sea otters, because of their local extirpation in Oregon, they're of interest to diverse stakeholders in our state. They're often cited as an ecological priority as a keystone species. They have top-down effects in kelp forest ecosystems. They eat urchins. And they keep those urchin populations in check, which keeps the urchins from eating too many of the kelps. So it keeps those kelp forest ecosystems healthy. And I think Bob Bailey is in the audience, so I'm going to give my shameless plug for the Alaka Alliance, who I am not affiliated with, but they are a group working to restore sea otters to Oregon. And they've recently published a feasibility study assessing the potential for sea otter reintroductions in Oregon. So I highly recommend checking them out if you're interested in learning more about potential reintroductions of sea otters to Oregon. And I also want to note that they work very closely with in partnership with Oregon tribes. And the original founder of the Alaka Alliance was Dave Hatch, who was a member of the Confederated Tribes of Siletz. This is a picture of the whale saddle effigy from the Macaw site of Ozette in Washington. And all these little white dots you see in the photo, these are sea otter teeth. So this was a wood carving of a whale dorsal fin or saddle studded with over 700 sea otter teeth in the shape of Thunderbird, who was a powerful non-human whaler and transformer. And even though this isn't from Oregon, I'm including this as an illustration of the cultural significance of sea otters to Indigenous peoples on the Northwest coast. In Oregon, sea otters often rank among the top marine mammals in coastal Oregon archaeological assemblages. We do know that sea otters were used for their pelts at contact. The Alaka Alliance runs webinars, and tribal members have been interviewed, and they state that they don't have evidence for dietary consumption or the eating of sea otter. And I've only seen a few ethnographic and historical references mentioning sea otter as a food, and they were very contradictory. So some folks said they were tasty and some folks said they were really terrible to eat. So we know that sea otters were important, and as a result, archaeologists have assumed that sea otters were used for their pelts, and there's this implicit assumption that tribal ancestors must also therefore have eaten the meat, but these assumptions have not been critically evaluated. So that question of tribal ancestral use is one angle of research that I set out to find some answers to, looking at these archaeological sea otter remains. So specifically the question of whether tribal ancestors were skinning sea otters to remove their pelts, to remove meat for dietary consumption, or both. And I also wanted to attempt to characterize what human sea otter relationships may have been like on the Oregon coast prior to contact. And just a quick side note, these data and results aren't published, so just please no photos where you see that icon. So to answer these questions, I looked at a lot of sea otter bones. I identified the sea otter remains in the Parti and Pomeroy sites. I looked at them with and without magnification to identify cut marks. This is a rib with some really fine cut marks under very high magnification. I took a lot of photographs, I made a lot of sketches, and then I compared my results to some previously published studies, including this sea otter cut mark study that was published in 2020. But just backing up a little bit, archaeologists have a long history of coming up with ways to understand animal processing in the archaeological record, largely by looking at cut mark type and location and then trying to guess the activity. So Lewis Binford, a really famous archaeologist, butchered a bunch of caribou and determined that heavy cut marks at major joints like the hip or shoulder were butchery removing the forelimb or hind limb. And finer cut marks along the length of the bones meant removing meat to eat. But sea otters do not look like caribou. So I compared my results to this skinning study as well as another study dealing with carnivores. And of course, small fur-bearing carnivores look a little different from caribou. Moss's study was really cool because she looked at archaeological sea otters from Southeast Alaska, but also incorporated an experimental archaeology component by observing Kyle Berry, a Tlingit hunter, skin this sea otter here. And in the course of skinning the sea otter, Mr. Berry left marks at locations that would normally be associated with butchery or processing for food, following, like, for example, Binford's rules. So she found cut marks in the hip joint at the femur and on the innominate and, let's see, on the ribs, on the fibula, but these were generated from skinning. which is sort of contrary to the typical assumption. So a really good methodological reminder that informed a lot of how I thought through my cut marks is that cut mark locations and their assumed functions are not necessarily universally applicable. So I identified almost 3,000 sea otter bones. I can spot a sea otter bone from about 100 feet by now, I think. Don't worry too much about the rest of the numbers. Just know I had a very robust sample size, although I did look at fewer bones from the Pomrose site. I do want to flag that forepaw bones or hand bones were extremely underrepresented or missing. The original excavators used 1/4-inch mesh to screen the dirt that they were taking out of the sites. And these are very small bones which would go through that screen. But forefeet are also sometimes removed with the pelt because they're such small bones, it's easier to just leave them in the pelt rather than work around them. These figures show cut marks from all the major long bones in the skeleton totaled on one animal for each site. So these are sort of all the Partee cut marks totaled and the Pomrose cut marks totaled for the major long bones. And the larger and darker the circle, the greater the number of cut marks in that location. So at Partee, the humeri and femora were most frequently cut, often in multiple locations. You can see all along the femur and the humerus. And the elbow, hip, and knee joints were really intensively processed. Again, you can see by the darker circles. And there was uneven distribution across the skeleton. And when we compare that to Pomrose, the innominate was most frequently cut, but only by a few cut marks. And compared to Partee, there's very even distribution across, across the long bones, 1 to 9 cut marks in a given location. So I'm going to walk you through a few examples of cut mark locations on these otters and the probable functions, which I determined by considering the sort of Binford conventional cut mark typology and previous carnivore cut mark studies. And this will give you some neat zoomed in photos too of the cut marks and the bones. So cut marks indicative of skinning or pelt removal included cut marks to the outer side of the mandible. This is very standard. You start by cutting at the mandible on the skull, and then from there you sort of work the pelt up and over the head and then move on to removing it from the rest of the body. So this is very clearly pelt removal. And I found these cut marks on juvenile sea otters as well, including pups. The distal tibia, that's your long shin bone. As you can see here, 1, 2, 3, and then some smaller ones here, because oftentimes you have to work around the ankle joint, and this is the start of the ankle joint. And cut marks to the tarsals, metatarsals, and phalanges are also very common locations for cut marks related to skinning, because again, you're continuing to work in the ankle joint to free the pelt from that joint. So this is an ankle bone. This is one of the metatarsals. Cut marks indicative of muscle removal or sort of processing of the skeleton, breaking it down into smaller pieces, included cut marks to the ribs and the sternebrae. The sternebrae is the sternum. We have one bone, that's a breastbone. Sea otters, it's made up of a bunch of smaller bones. And there's lots of ribs on them, a lot of meat on the ribs. And then the vertebral processes, which face outwards along the spine. There's a muscle called colloquially the backstrap, which sort of sits here next to the vertebral process. And then this is the inner surface of the vertebra, so sort of facing the internal organs. So there's no way these cut marks could be associated with pelt removal. This is some sort of processing, maybe organ removal. And then I had cut marks that were quite frankly ambiguous or maybe reflected multiple functions. So the radius and ulna, the bones that make up the lower forelimb and the elbow joint, Moss in that skinning study, she noted that the sea otter's forelimbs were drawn in towards the body with rigor mortis, and it was really difficult for Mr. Barry to remove the pelt from them. So I hypothesized that maybe these cuts reflected pelt removal, trying to remove it from the elbow joint. The radius and the ulna are also kind of perfectly sized for tool manufacture. They're very strong and compact. So maybe the bones were being removed at the elbow, sort of as a raw material for tool manufacture. And then cuts to the hip joint, the innominate, the femur, probably a combination of skinning like moss found and or meat removal or taking apart the hind limb. So these are the cut mark locations on the skeleton grouped by probable function. So we have those that probably reflect skinning or pelt removal, those that probably reflect meat removal, and those that were a little unclear. And I wanted to try and evaluate in some way how likely it was that meat removal for dietary consumption was occurring. So I turned to a meat utility index, and a meat utility index ranks how much meat by weight can be derived from different parts of the skeleton. So the femur has X pounds of meat yield. I won't walk you through every single element, but for example, these humeri, radii, and ulna, these highly cut marked bones, they're actually ranked very, very low in the meat utility index, yet they're very well represented at the site. So if they were used for meat for dietary consumption, they wouldn't have yielded all that much. And then conversely, the vertebrae and ribs are ranked first and second in the meat utility index. They would hypothetically yield the most meat in terms of weight, but these bones are actually underrepresented at the site. So again, possibly not yielding all that much meat unless they were cooked and disposed of elsewhere, but I don't have any archaeological evidence for that to my knowledge. So I sort of had to work through the logic of all these rankings and how many of these parts, how many of these bones were present at the sites. And I sort of settled on the conclusion that innominates, femora, the vertebrae and ribs probably had meat removed, but based on that underrepresentation of like the vertebrae and the ribs, they may not have yielded large quantities of food. So sea otters may have been consumed occasionally, but they may not have been a dietary staple. Other food sources like sea lions, seals, whales, elk, very large, and in the case of the marine mammals, fatty with all that blubber, they would have been available year-round or seasonally for the inhabitants of Seaside, and they're also present in the Parti and Pomerose animal remains. And you can sort of, like, that's a sea lion right there and a whale, so lots to choose from. And then an additional possibility is that at both the Parti and Pomerose sites, there's evidence for carnivore gnawing on the sea otter bones. You can see these punch marks here. These are made by canine teeth. And we know at contact dogs were important elk hunting partners. So perhaps sea otters were hunted for their pelts and then processed for feeding dogs. And some of these gnawed bones were found very close in proximity to the remains of a house structure, particularly at the Pomerose site. I also want to touch on this question of human-animal relationships. There's a tendency to evaluate archaeological animal remains in terms of function, as I just did in terms of pelt and meat. I mentioned earlier that the forepaw bones were underrepresented, and this could be a result of archaeological recovery techniques or forepaws being removed with pelts, but maybe paws were removed for use as amulets. Or other animism-related reasons, although I make that suggestion with a lot of caution. I'm a Euro-American writing about a community that I'm not a part of. And again, another example of sea otter use outside of Oregon and the macaw whale saddle, this link between sea otter as an indicator of wealth, status, or chiefly prerogative. Sea otters appear in Oregon tribal oral histories and traditional narratives. In the story, The Invisible Husband, told by Clara Pearson, there's a specific reference to Seaside as the location where all those men went sea otter hunting. Sea otter pelts are guests who attend the moon's winter dance, along with guests like Dentalium. There's a story about a woman marrying a sea otter, and she and her husband send a beached whale ashore each year as a gift to her family. So these accounts are by no means comprehensive, but they're indicative of human sea otter relationships beyond an economic function. Sea otter pelts were really powerful items associated with wealth, sometimes like with a shaman's powers. So the persistence of these relationships in these sort of oral stories definitely illustrates their significance, I would argue. So in conclusion, the sea otters were skinned for their pelts. The meat was probably occasionally eaten by humans or maybe fed to dogs. And sea otter bone was used for tool manufacture, like these sea otter bacula here have all been turned into awls. I want to emphasize that while this research is a new contribution, these results are not a discovery. I'm building upon current Oregon tribal knowledge, and I sort of propose my research as a line of evidence affirming that knowledge. Despite the extirpation of sea otters from Oregon, present-day tribal members maintain a really deep knowledge of and connection to the sea otter. And I recommend searching out Indigenous voices on this subject. Check out the Alaka Alliance and their articles or videos from folks like Peter Hatch, for example, and see what the Oregon tribes have to say about possible sea otter reintroductions and Indigenous peoples in Oregon. So now I'm going to move on to a more biologically applied angle of my research, and that is studying the ancient DNA of the archaeological sea otters themselves to learn about how the now locally extinct Oregon sea otters relate to living populations. And this relates to potential future reintroductions, because when you are reintroducing a species to an area, you want to be sure that the source population you are using for reintroductions are a good match physically, genetically, et cetera, with the original population that was lost. So this research was published last December, and it answers our specific research question: do the archaeological Oregon sea otters share greater genetic similarities compared to modern Alaska or California subpopulations? And to answer this question, we analyzed the mitogenomes from the Seaside sea otters and compared them to mitogenomes from California and other northerly sea otters. And a mitogenome is the mitochondrial DNA, or the genome of the mitochondria in the cell. If you think back to high school biology, the mitochondria is often referred to as the powerhouse of the cell. But the really important thing to remember is that this is inherited from mother to offspring, so it reflects the maternal line only. And Oregon has been proposed as a transitional zone for southern and northern populations. Previous studies, including other ancient DNA work by scholars like Sean Larson and Kim Valentine, have demonstrated that there's a latitudinal climb, or a change in physical and genetic traits of sea otters as you move north from California to Alaska. So based on previous studies showing this climb and the far north location of the Seaside sites in Oregon, we expected the archaeological Seaside sea otters would share mitogenome lineages with sea otters from northerly latitudes. So we obtained mitogenomes from the tooth dentin, which is the bony part of the tooth root you see here, from 20 of the archaeological Seaside otters. And we obtained mitogenomes from dental calculus from 21 historic and modern sea otters in the National Museum of Natural History Mammals collection. And dental calculus is this buildup here. The plaque that the dentist scrapes off your teeth at a cleaning can, if it's not removed and it builds up over time, it becomes hard and it mineralizes, and it traps microbes and bacteria, but also some of your own DNA, because it sort of is formed as part of an immune response. So we call that host DNA. So this dental calculus here contained microbial DNA, but also the DNA of this particular ancient sea otter. And this is actually a really nice way to do an ancient DNA analysis, because it's not destructive. You don't have to drill into anything. You just sort of pop off the dental calculus. And lucky for us, sea otters don't go for annual dental checkups. This is a map showing the geographic origins of our samples. We tried to sample as best we could from across the Pacific Rim. So we had a sample from Japan, a few from Russia, Alaska, the Aleutians, British Columbia, Washington. We were really fortunate to obtain 2 really rare pre-fur trade otters from Oregon, which was very exciting. And then for California otters, we used previously published mitogenomes from a 2017 study, as well as 5 historical dental calculus samples from the Santa Barbara Museum of Natural History. So museums as repositories were definitely our friend for this study. I collaborated with Dr. Courtney Hoffman at the Laboratories of Molecular Anthropology and Microbiome Research at the University of Oklahoma. Ancient DNA extraction and sort of more generally lab work is performed in a really sterile, clean environment. As you can see in this photo, we were masking up even before the pandemic. We were making it cool way back then. We performed tooth and dental calculus sampling, and we extracted the ancient DNA. And we used a process called mitogenome capture. A good analogy is it's kind of like fishing. You use sea otter specific primers or baits to grab just the sea otter DNA, and then you sequence those DNA fragments and put them together into the full mitogenomes. And we then funneled those mitogenomes into a network analysis, which is seen here. And a network analysis shows the relationships between what we call haplotypes or a given mitogenome sequence. So the size of the circle is representative of the number of samples that share a given haplotype. And then these little hash marks represent the number of differences between two haplotypes. So you can see haplotype 25 and 24 only have one difference between them, whereas haplotype 14 and 23 are showing quite a few differences. And I'll walk you through this to sort of show what this all means and what interpretations we got out of this network. So first, here in the black circle, you can see the modern and historic California haplotypes. This represents a total of 48 individuals, but we only have 5 haplotypes. In comparison, the 20 archaeological sea otters from Seaside yielded 10 haplotypes, which are circled in red, and 5 of those haplotypes are unique to a single individual. So in terms of just haplotype count, this suggests greater genetic diversity in the archaeological otters, which is expected given the population bottleneck caused by the fur trade. And a population bottleneck is when a population loses so many individuals that it loses a lot of its diversity. And generally speaking, in wildlife conservation and biology, greater genetic diversity is a good thing. And you can really see sort of the impacts of the fur trade when comparing the pre and post fur trade haplotypes. In terms of that latitudinal climb I mentioned, or the changes in genomes moving from south to north, there's a very clear separation, or a sort of a large distance, if you will, between those California haplotypes and sea otters from northerly latitudes, including the archaeological Oregon sea otters. So evidence for that latitudinal climb in our mitogenomes. This is a zoomed in view of the northern haplotypes in the network analysis. This group of haplotypes circled here contains 7 archaeological haplotypes, and most of those are relatively close to the historical Oregon, Washington, and British Columbia individuals. This circle contains Alaska specimens, which are particularly close to 4 of the Oregon haplotypes. The Russia and Japan samples are relatively distant, but the Russian sample is quite close to one of the archaeological Oregon specimens, and a previous study by Sean Larson, they were looking at microsatellites, but they also found gene flow with Russian sea otters. So it's neat that this signature showed up in our study as well. Something else to consider, because we're working with archaeological sea otters, is the issue of chronological time. The Pomeroy site's older and the Partee site is slightly more recent. So we performed a TempNet analysis, which visualizes shared haplotypes between chronological periods. So here we have 2 haplotypes, which are shared between Partee and Pomeroy individuals, which suggests the persistence of some mitochondrial lineages through time in the Seaside area. And we suggest that these overlaps are consistent with local sea otter hunting, because Partee and Pomeroy are so close geographically and in age. And I mentioned earlier, Clara Pearson described Seaside as the place where all those men went sea otter hunting, which to me is a really nice parallel between the oral histories and then the archaeological evidence. So this is a screenshot from the Alaka Alliance feasibility study. And I'm including it because it turns out that our ancient DNA study ended up really fitting nicely in with the existing body of research in previous studies, particularly with regards to showing the existence of the Oregon coastline as that latitudinal cline. And in the feasibility study, the authors assessed the current body of DNA evidence around Oregon sea otters, including our study, and they concluded the original Oregon sea otters likely represented a hybrid zone with ancestors that genetically resembled both southern and northern sea otters. So I found that very exciting that all these studies have ended up pointing to sort of a unified conclusion as the feasibility study authors analyzed it. So some concluding thoughts, bringing all of this together. I didn't necessarily state several of these points at the outset, but I hope that I have illustrated how biological and ecological aspects of animal remains from archaeological sites can be linked with cultural beliefs and activities that resulted in their deposition in archaeological sites by tribal ancestors. That there's this sort of reality that animal remains from archaeological sites represent both tangible and intangible cultural heritage from the past. And these animal remains can inform both the biological and cultural aspects of conservation efforts. And finally, while archaeological excavation is probably the first thing we think about when we imagine archaeology, and it's certainly an exciting aspect of the profession, there are archaeological remains that are curated in museums that have not yet been studied. And these are sometimes called legacy collections because they've been with a museum since their excavation many, sometimes decades previous. And these museum collections hold really important data that's waiting to be analyzed and released into the world. So this is my pitch for working through those backlogs of data and seeing what they can contribute. I'd like to thank the folks listed here, especially my University of Oklahoma mentors, co-authors, and colleagues, and my funding entities, including the University of Oklahoma and the National Museum of Natural History, Robert Heavey and Constance Spilling Fellowship, without which both projects I've discussed today would not have been possible. And thank you all for your attention.
That was so fascinating. I learned so much. Thank you for sharing that, Hannah. So folks, if you have any questions, please add them to the chat or Q&A. But I'll kick us off with one. Hannah, I always like to ask, what was your inspiration behind selecting, this specific topic and kind of area to study a bit deeper in?
Yeah, it really was Dave Hatch, who I unfortunately didn't have an opportunity to meet. But I started my master's sort of before the Alaka Alliance got fully revitalized. And I knew that sea otters were really important to Dave Hatch. And I thought, maybe I should pursue that. And I was really also sort of just blessed with this archaeological collection because it's so well preserved. So there are another 9 million dissertations in this collection. And so I thought, wow, pick a marine mammal and let's pick sea otter and see if I can be of use.
Yeah, totally. I find the sea otter fascinating, but I really was in awe over the DNA research and to find that Oregon sea otters are a mixture of both. So I'm very curious to see, like during reintroduction, what does that mean as far as, reintroducing them from where?
Yeah.
So that'll be fun to see play out, hopefully.
Yeah.
So one question, how does someone from Oklahoma connect to Oregon's otters?
So that was actually, I worked at the Smithsonian National Museum of Natural History before I started my master's and PhD, and Courtney Hoffman, who was my mentor and colleague for the ancient DNA, she was at the Smithsonian doing her PhD at the same time. And so as it sort of is in the wonderful world of academia, you stay in touch with your colleagues, and we were at a conference and she said, hey, maybe we should look at this. And I said, wait, could we really do that? And we sort of ran with it. And off we went. So I got to spend 2 sort of summers and falls in Norman, Oklahoma. Boomer sooner.
So is that where you learned? Because throughout the presentation, I'm like, wow, she must have had to take so many anatomy classes and learn what kind of cuts. Did you learn a lot of that through coursework or through your experience?
It's really through experience. A lot. I took a zooarchaeology class in college, and that really sort of gives you the basics. But then after that, it's sort of exposure and practice makes perfect. And sometimes I'm working with undergraduates and they go, but how do you know? And it's like, well, I've had 8, 9 years of practice just looking at bones. My brain is sort of a 3D Rolodex at this point, but we do have weird amounts of anatomy. I know some weirdly specific things, but the ancient DNA was very much, you have to learn the proper lab protocols and all that jazz. So that was a really fun but huge learning curve.
So remind me, did you get all the bone specimens from previous collections, or did you also go out to some of the sites and get your own?
All previous collections. So I didn't do any field. I've done field work during graduate school, but not for my actual PhD. So it was all working with those museum legacy collections.
Fascinating. Let's see. Here's just an FYI that a sea otter has been observed at Yaquina Head, Newport the last few days. Oh, thank you.
That's exciting.
Yeah.
Yeah, they do float down sometimes from Washington, but until it's a whole bunch of them establishing a breeding colony, it unfortunately doesn't count, but that's exciting that they were hanging out there.
Yeah, that is. Oh. And with that, I think that's all the questions for now. And so can folks go somewhere to find out about the whale research that you're also working on?
Sure, I am. Let's see. Actually, not yet. That is, I need to give a public talk on the whales. If they're interested, they can certainly email me. But I am working on getting that all written up in a palatable, readable version that's not dissertation formatting to be published. I have had some previous whale work written up in the Hakai magazine. If they want to go check that out there, because I've also done previous articles on these whales as well. So
nice. And then one more question came in. Can you tell anything from the teeth regarding food sources for the early otters?
So from looking at them, it's tricky. There have been some studies that have shown you can try and age a sea otter based on the wear, because they can be pretty tough on their teeth because they're just chowing down on all sorts of different things. But I can't look at the teeth and visually say, oh, they were eating a lot of, that's not true, I take that back. You can look at the canine teeth and tell if they were eating clams because they hook it in their teeth and it creates this really distinctive wear pattern on the back. So it's kind of like they're using their teeth like a bottle opener over and over and over. And so it wears the enamel at the back. But for the back molars, it's hard to look at it visually and just know, other than that they're old and that they've been sort of wearing their teeth down for a while. But you can do, again, sort of minimally destructive sampling of the teeth, and then you run the isotopes, and that can tell you sort of where they are in the trophic cascade and what they're eating. And I'm actually working on a paper with a colleague right now, looking at that.
It's amazing, all the little details.
Yeah.
Here, there's a few more questions. Are there extinct genes, and would you advocate for a reintroduction by way of CRISPR?
Yeah, so mitogenomes, you don't really get a view of specific genes the way you would like a full nuclear genome. There is a paper out by a researcher named Annabelle Beichman. She used to be at UCLA, I think. And if the person asking that question is interested, I would recommend looking at her work, because she was, I believe, looking at specific genes for something like pelt thickness. That's a very oversimplified example. I don't do nuclear genomic work. But I imagine that other people will be looking at sort of nuclear genomes. I can't speak to that with my particular research, but I'm sure there will be some research in that direction soon.
And is there a stable isotope signature for sea otters that would allow to see if they are in human diets in the past?
Yes, you could compare it to a human.
Okay.
Or sort of like generally think about what humans would be eating, and then you can sort of map sea otters onto that. I haven't seen our results from the stable isotopes yet, so I can't quite tell you the answer, but yes. And there are a lot of archaeologists who look at that, not just with sea otters, but with like sea or like dogs. It's a really common thing to look at with dogs, to see if humans were provisioning or feeding dogs the same things that the humans were eating, or if they were giving them sort of like the bad fish, that sort of thing. Sort of how well are your dogs eating?
Yeah.
But yes, that is something you can look at.
How do you think studies and results like yours can best educate and influence cultural narratives and scientific representation around otters that the primarily settler population of Oregon has?
Again, I would not presume to speak for any tribes with my work. Like I said, I'm sort of delivering this to the Alaka Alliance and emailing it to tribal members saying, hey, here, if you can use this, please do. But I think anything that can emphasize, I used to volunteer at a tribal museum in the state of Maine. And the number of people from settler colonial descent coming through the doors and being quite perplexed that there are native tribes still here. If we can emphasize that really sort of these tribes have been here since time immemorial, and they are still here, and they have held these connections with the landscape and with the animals throughout all that time. And emphasizing the presence and all that the Indigenous peoples of the world still do for sort of the benefit of the environment, et cetera. I think that would be a good thing.
Yeah, absolutely. All right, Hannah, thank you so much again for presenting for us this evening, and thank you everybody for joining us. Hope to see you again at a future presentation, and I hope you all have a wonderful evening. Good night.
Night.